So the next speaker is Luna Yamamori. She comes from the Faculty of Agriculture. The title is a study about the twinkle of fading of Aurina aurita. So please start your presentation.
Thank you for introducing me. Hello, my name is Luna Yamamori. I'm a student of Kyoto University, learning marine biology. I studied about the trigger of feeding of a jellyfish named Aurelia. I know jellyfish very much. So this is the outline of my presentation. I will introduce my study around this. So Aurelia is called in Japanese Mizukurage, the most famous jellyfish in the world. The main character in this presentation is Flip. This is Flip. Isn't he very cute? I love it very much. Sorry, I came here with Flip. And this is Flip mouse and Flip is like a baby jellyfish. Importantly, they eat plankton. So, as we get a movie of Flip's feeding behavior, see, a needle will touch Flip's arm and Flip constrict and open mouth. Now, could you see? So, I saw this every day and wonder, how do Flips distinguish food from unedible things? There are many unedible things floating in the sea. So if Flips reacts to everything touches the arms, it uses needless energy. So about that, there's a previous research done by the American researcher. He used Cordillera, another kind of Flip, and revealed that Cordillera distinguishes food by particular kind of amino acid. But about this, I wonder, do Flips really distinguish food only by amino acid? We humans can distinguish amino acid too, but we can also distinguish sugar. Four cases are delicious, isn't it? So, to examine this, I did the first experiment. In this experiment, I exposed Flips to peptone, a fragment of protein, and glucose, sugar, both in 3 concentrations. This is the result. The blue bar represents the number of Flips getting to peptone. And the concentration decreased, as you see right. As the concentration decreased, the blue bar shortened. But in all densities, Flips reacted. And on the other hand, there are supposed to be red graphs, but there isn't, which means no Flips reacted to glucose. For that, let's answer
for your presentation. So is there any question or comment? Please.
What is your motivation of doing this research?
Thank you for your question. As I mentioned, I saw breastfeeding behavior every day. Like every day. I have a question. How do breeds distinguish food? That's the motivation. Does it answer?
Maybe. Second question is that how do we human beings distinguish food from uneatable thing?
So,
Maybe the very small children can distinguish. How do you think? How they learn?
so it's my opinion, but I think it's taste. Children don't eat so bitter things, and children don't eat iron. It's taste, I think.
Taste.
Taste.
You need to get it into your body, but it may be not a good thing. So before taking taste, you need to distinguish food from bad.
I understand.
Okay, thank you very much.
Okay.
Yeah, okay.
Sorry. Thank you.
Thank you. Also, please.
I just have a short question. You said that the polyps are not distinguishing glucose sugars. Can I ask, under normal environmental conditions, does seawater consist of any glucose sugars?
Thank you for your question. So about this, I can't say for concrete precisely, but I think a little.
No.
Thank you.
Please. Yes.
I want to know. Sorry.
Okay.
I want to know the amino acid sequence of I want to know the amino acid sequence of peptone and fermenton. What is the main difference between these two?
Thank you for your question. Well, the main difference between them is membrane. Do you know membrane? Thank you. It's a skin of cell, and both plankton and fermenton are covered with membrane, but peptone is not. So I think, so brings react to the particular the amino acid, particular in membrane.
Maybe
Is
the,
it answer?
yes, maybe the difference can help you to answer the second question to search the
Yes,
key amino acid.
yes, I say it's key.
Thank you.
Thank you.
Please give your question.
Thank you for your presentation and cute jellyfish pictures.
Picture.
Yes, thank you for your interesting presentation and cute jellyfish pictures.
Do you mean you want to see the picture of jellyfish?
No.
Sorry.
A brief question. Please ask you to. Sorry. I wonder why did you try? Why did you think that what can be the trigger? For example, some lives like fats. For example, human beings like fats. So, why
why do you think what can we trigger and try to
So I'm sorry, but I'm afraid we don't have enough time for this question.
Sorry.
I think you can talk about with her after this session. We are sorry about that.
Please come and talk to me after this session.
Yeah.
Thank you. Thank you.